Right, following on from PCR, we're now going to have a look at some of the uses for this DNA that we have copied using a process called gel electrophoresis. So gel electrophoresis is a process that we can conduct in order to separate out DNA of different sizes in order to help us to produce something that we call a DNA profile or a DNA fingerprint. So I've got a very basic setup for gel electrophoresis that you can see here in the diagram. There are some important things that I want to note for this as we get further into this lesson. The first thing is, in gel electrophoresis, we have a agarose gel, and this is represented by the blue rectangle that we have here. And this agarose gel, at one end, we load in our DNA samples. So this is in this sort of green color up the top. And we load them into what we call these wells. Now, what you'll notice on the left, we have this molecular marker. This is sometimes also known as a DNA, oh, I've written that completely wrong, a DNA ladder. And this ladder is useful as sort of a key for us to identify DNA sequences of particular lengths. Now you'll notice as we run down the left, if I highlight molecular marker and ladder, you'll see that we have 1,700 base pairs, 1,000 base pairs, 500 base pairs, 200 base pairs. And hopefully what you're noticing is that as we get further away from these wells, these DNA sequences, these portions of DNA get considerably smaller. Now, what tends to happen as we run gel electrophoresis as a process is DNA moves through the gel, and I'll talk about how it does that in a second, from the wells to the opposite side of the gel, and it separates it out based on its size. Okay, so those that don't move very far through the gel end up along the top, okay, closest to the wells. Okay, so these are our largest pieces of DNA, and those which move really well throughout the gel, let me highlight this maybe in a different color, so our largest is in pink here. Those that move really well through the gel, that are really small, end up being our smallest DNA fragments. So how does this all work? Well, nice and simply, we know that DNA itself has a slight negative charge. And this slight negative charge occurs thanks to the phosphate group that forms up the nucleotide. Now, for the chemists or the physicists out there, you'll know that a negative charge is repelled by things with a negative charge. And so what we have is we have an anode and a cathode attached to our gel and to our solution that's in the gel, so that buffer solution. And what we have is that the negative DNA is repelled by the negative electrode, and it is attracted to the positive electrode, which is positioned at the opposite end of the gel. And so DNA is encouraged to move through the gel, but unfortunately the gel is only slightly porous, meaning that large pieces of DNA will not make its way through many of these pores, getting stuck at the very beginning of the gel. However, smaller things can navigate those smaller pores and will make it all the way through the gel, allowing us to distinguish things based on different sizes. So, in order to help us understand some of the how involved in this gel electrophoresis, instead of just sticking entire lengths of DNA in there, we need to talk about restriction enzymes. Now, we've spoken about restriction enzymes previously, but we use them just following PCR and we add them to our DNA in order to cut DNA fragments of specific base sequences and to cut them at specific recognition sites. So, for instance, EcoRI, so Escherichia coli RY13 is our source for this one. It cuts at a very unique source, or very unique recognition site, which is GAA-TTC. Okay, we can look at TAC-I, so again, the source being Thermus aquaticus, it's going to cut at TCGA. Now, what happens here is they all produce DNA fragments of different lengths, depending on the actual base sequence of the DNA itself. So here are a couple of key components for our gel electrophoresis setup. I'm not going to go through these too much right now. The only thing I really want to draw attention to is something that I haven't spoken about in great length now, but it is that you're surrounding this entire setup within the actual electrophoresis equipment. We do have a solution, okay? And this solution allows for the flow of electrons through this gel, and it encourages the movement of DNA throughout the gel electrophoresis setup. So as a quick reminder, if I get rid of that highlighting there, we look at the negative terminal is A, so that's going to end up on the edge with our wells, and then our positive terminal, what the DNA is attracted to, is down the other end here, okay, being what our DNA is then attracted to. So when we have a look at our ingredients for gel electrophoresis, in the same way that we had ingredients for PCR, same as PCR, I'll highlight now, we have a DNA sample, okay, this is what we've amplified during that PCR process. Unique to gel electrophoresis is that agarose gel, so that gel that is porous, allowing the DNA to move through it. We also have a buffer solution. Now, a buffer solution is not unique to this one, but as we mentioned in PCR, it allows for the maintenance of that ideal pH, and it also provides some ions for conductivity of that solution. We then move down into one of the more unique things. We have some electrodes, and as we've mentioned, we have the positive red and negative black electrodes. So the positive electrodes are sometimes referred to as cathodes, and the negative electrodes are sometimes referred to as anodes. Just something to make sure you're aware of in case it pops up throughout the rest of this course. Finally, we have some wells, and we have that fluorescent binding gel, okay? And what this allows is for the DNA to have a place to start, and it also allows DNA to be viewed in order to analyze the bands that we get at the end. So, we've previously mentioned this marker, or this DNA ladder that runs down the left-hand side, and as previously mentioned, we said that it contained fragments of known lengths, and those known lengths allow us to serve as a comparison point for the DNA samples that we get. So if we were to have one particular example, so we see sample A, I'll highlight it in green now. Sample A only has one band, and we can see that that one band lines up with the 1000 base pair marker. So we can determine based on this DNA ladder, using the DNA ladder, I should say, we have sample A has 1000 base pairs in length. So how does gel electrophoresis work? I'm sorry, this is a little bit wordy here, but let's simplify it and go through it nice and quickly. So firstly, we isolate DNA, we amplify it using PCR, we use a restriction enzyme to cut it into small pieces, and then we place those pieces into the agarose gel. An electric current is applied to the gel, and it's going to pull the fragments towards the positive terminal, okay, of that gel, away from the negative terminal. Why? Because the DNA is negatively charged thanks to the phosphate group. Fragments are going to move at different rates depending on their size, where smaller fragments move faster or further, and larger ones move less and move slower. And what we're going to see as a consequence of all of this is banding representing different DNA fragment sizes. So, how are they all separated? As we've mentioned, the DNA is negative, it's pulled towards the opposite end of the gel, which is positive. And these different rates help us to separate out into fragments of different sizes that are unique. Okay, so let's look at the relationship very quickly. I quite like this graph as a representation of the relationship between how big these fragments are and how far they move. So things that move the most, okay, across the x-axis, and their size on the y-axis. So we can see that it moves significantly further the smaller that these fragments of DNA are. So what are some of our uses? Firstly, similar to PCR, we can use it to identify hereditary disease. We can use it for testing for genetic disorders, early diagnosis of some diseases. Interestingly, we can use it in paternity testing and forensics, which is what we're going to have a look at now. And we can also use it, critically in this course, as evidence for evolution in tracing some ancestry. So let's look at one example together now. Okay, well, I've got some previous highlighting in here. We'll just get rid of that very quickly. So when we're having a look, this is an example of paternity testing where we have a mother and a child and we're trying to determine who the father of that child is. Now what we can see is that the mother shares particular DNA sequences with this child, particular genes of interest with the child, but it doesn't explain all of the child's genetic features, okay? So when we're having a look through, we know that this child has this band here, and we can see that, okay, dad 1 doesn't have this, however dad 2 and dad 3 do have this same DNA marker. We then have a look down a little bit further and we can see, okay, dad 1 and dad 3 have this marker, but not dad 2. And finally, for the third little piece of info, we can see that dad 2 and dad 3. Now, based on this info, we can kind of use it to identify that, okay, it's unlikely that dad 1 and dad 2 are the father of this child, and that the only possible explanation for all 3 of these particular genes of interest would be dad 3. So one final example, and again I've done some highlighting previously here, is we've got now a crime scene and we've got a victim. So if we have a look at the victim, we can see that the crime scene, that the victim's DNA has been found on the crime scene, but it doesn't explain everything in the crime scene, okay? So what we can have a look at is we can have a look at the 3 suspects involved in this crime, and we can see whether or not their DNA is found throughout the crime scene. So first of all, we have a look at the crime scene and we look at this first piece of evidence. We can then have a look at the crime scene for the second piece of DNA evidence. If we highlight all that all the way through, we can then have a look at the fourth piece of evidence, or third piece I should say, and finally the fourth piece of evidence. Now, the only suspect with all key pieces of evidence in this instance is most likely to be suspect number 1, okay? Now, suspect 1 again has the large majority of these pieces of DNA, and suspect 2 could also be involved. Suspect 3, very unlikely to be involved.